Uh, Practical exams for microbiology, pathology, and pharmacology are starting next week for both NTR and RGHS universities. The practical exams consist of 80 marks practicals and 20 marks viva. Gram stain, Zealands and stain spotters, applied bacteriology, virology, mycology, and viva. 4 examiners, 5 marks each. The first training you are supposed to do is gram staining technique. It is to differentiate between gram positive and gram negative organisms. Cover the smear. The trick is you have to identify which side smear is. Most of the students stain on the reverse side and they break the slide. Don't do that in the examination. Cover the smear with gentian violet. Leave it for 1 minute. Wash with water. Cover the smear with gram iodine, mordant, and wash with water. Decolorize with acetone or alcohol drop by drop, and cover the smear for counter stain. The counter stain is safranin or dilute copper 16 for 30 seconds to 40 seconds. Wash, blot dry, and see under oil immersion microscope. Adjustment of microscope, fully raised, and 100x objectives. You have to touch the smear where the oil is put. Raise it up until you something, and write neat round circle in which you have to write the gram positive arranged in clusters resembling staphylococci and gram negative bacilli are seen. Once you complete the exercise, raise your hand, call the examiner. Examiner most commonly asked question is mechanism of gram reaction, theories of gram positivity, electrical, mechanical, and chemical theory. Electrical theory, gram positive cytoplasm is acidic and affinity towards basic dye. Mechanical, gram positive cell wall is 40 times thicker than gram negative, and gram negative cell mostly made up of lipoid layer. Alcohol dissolves the lipid content of the gram negative cell wall and it is thinner. The iodine complex comes out and takes up the counter stain. Two chemicals, magnesium ribonuclease and teichoic acid, are also responsible for gram positivity of organisms. They will ask you gram positive cocci and gram negative bacilli, gram positive examples. Example of gram positive cocci are staphylococci, streptococci, and pneumococci. Staphylococci is gram positive cocci in clusters. Uh, Streptococci is chain, gram positive cocci arranged in chain. Pneumococci lancet shaped, broader end facing each other. Two cocci enclosed in single capsule. Neisseria, meningococci, plano-convex, half moon, plane surface facing each other. The gonococci kidney shaped, majority of them intracellular, kidney shaped, concavent facing each other. Bacilli, gram positive bacilli are remember as cell, CELL, Corynebacterium, Erysipelathrix, Listeria, Lactobacilli, then Clostridium, cell can, mnemonic, Clostridium, anthrax, then actinomycetes. These are all gram positive. Gram negative are mainly enteric bacilli. Remember E. coli, Klebsiella, SSPP, Salmonella, Shigella, Proteus, Pseudomonas, Vibrio, Spirochetes, and gram negative Coccobacilli are given as Netobacter gram negative bacilli, Enterobacter gram negative. The gram negative Coccobacilli are remember with mnemonic BBHY, Bordetella, Brucella, Haemophilus, Yersinia. Rest are all, most of the organisms are gram negative. So now clinical relevance of gram staining. Gram positive are susceptible to beta lactam antibiotics. Gram negative are more drug resistance. They need aminoglycosides or sulfonamides and quinolones are more difficult to treat. And once you see a gram stain, it's a gold standard in microbiology. You can call the clinician and start what attending whether it's monoglycoside or cephalosporin. If it's gram positive, it should be beta lactam. If it's gram negative, mostly it's aminoglycosides. If it is pseudomonas, anti-pseudomonas, gram negative, it's also gram negative. You can anti-pseudomonas, penicillin, piperacillin, tazobactam is drug of choice. Gram variation, when gram positive organism looks gram net, is called gram variation. Usually old culture, when it grows bacteria and the penicillin grow media containing penicillin, which spheroplast and protoplast, they are round in shape. They are called L forms because they are discovered in the Lister Institute of London. So these are the questions that is commonly asked in your gram staining. All of them you have prepared for this gram staining, and now in NTR it carries 20 marks, and there is no uniformity in the allotment of practical marks. It varies from, it is different in deemed university and RGHS and NTR. In NTR it almost carries 20 marks, and definitely you should score more than 15 to 16 marks. That's one of the important exercise. If you are not done, there is still time. You can go and practice this gram staining technique in your go to the department. They'll give you a slide and stain and try to focus and show it to one of your teachers. Thanking you.